My name's Prabs. I'm from Genomics England, and I am Director of Clinical Data, but I'm also a pathologist by training. So I've got 10 minutes to try and talk about our work on tumour whole genome sequencing and the value of linkage to healthcare data. We've been around for 10 years, so actually that's quite a lot to get through. So what I'm going to try and do is we've got a publication that's coming out in the new year. I'm just going to tell you a little bit about kind of the insights that we garner from that, what we're talking about, and the value of whole genome sequencing in cancer. And then also kind of the ask from Rory at the start or this morning is what would we like? And I'll tell you a little bit about what I would like to see anyway. So next. Yeah, okay, fine. So I'm starting with the very large assumption that most people know in the audience who Genomics England are, but for those of you who don't, we're actually a government-funded organisation that started 10 years ago now. The aim was to establish basically standardised high-throughput whole genome sequencing, or WGS, for patients with cancer and rare disease. And the aim was also to develop an automated ISO-accredited bioinformatics pipeline. So in effect, what we could return to the NHS was a clinically accredited variant calling and variant prioritisation as well. But in that, participants, or patients, but we call them participants, were also offered a research consent. And the ask here from them was to allow their genomic data to then also be linked to longitudinal health records and ultimately then be shared with researchers in an anonymised fashion in something that we call our trusted research environment. So all of our cancer patients had two genomes. They had a germline, which is their blood, and then they also had their tumour sequence or the somatic genome as well. And we have about 17,000 cancer patients, and they scan about 33 different cancer types. Although when we registered them, we also collected quite a kind of broad research type data set. Patients evolved, tumours evolved, diagnosis evolved. So actually we realised quite early on that we had to link to longitudinal registry type data sets. And we thought there's a value to kind of developing a real world type data set. So we have relationships, much like UK Biobank, with the National Cancer Registration Analysis Service, which is called NCRAS. And that data set contains cancer registration data, things like histology, SNOMED codes for that as well, and also systemic anti-cancer therapy, or SACT, which is chemotherapy type data. And the other bit that's also there is hospital episode statistics, or HES, and we also collect mortality data from the Office of National Statistics. The aim for us was really to kind of advocate that whilst you had these genomes, but by linking to the registry type data, that we could really understand and develop our genomic research, and also discovery could then be fed back into healthcare as well. So by linking at scale our registration data to the National Cancer Register, we could put together really rich insights on our population for cancer at scale. So the graph on the left is basically taking some of our tumour subtypes and actually profiling by stage of each of those cancers. We could also really kind of look at when that sample was taken in relative time to when they were in their treatment. So were they treatment naive, had they had their first dose of chemotherapy as well. And that's at scale, but also what we could do, and what's really important in these kind of ultra-rare cancers, for instance, is that we could link that and kind of develop patient journeys. So we could see episodically for each patient when they had their diagnosis, when they had their first operation, when they had their chemotherapy. So that was a real insight that we could garner from the National Registry and the healthcare data. But the other bit was also is to standardise all of this data that we had. UK Biobank trailblazers many years ago that they developed a mapping system for HES. So at the moment they conformed all the HES registry to a common data model called OMOP. And we've basically followed that as well. The other thing that we've done now is taken the NCRAS, or the National Cancer Register data, and also mapped that to OMOP mappings as well. And as part of our publication, we'll also be sharing those mappings as well. So we've now had quite a standardised protocol for genomes. We've collected some registry type data. What did we do with it, and what have we got insights from it? So this is quite a complex table, and I'll just try and go through it as simply as I can. So NHS England at the moment provide a test directory for cancer. Basically, it's what it's saying is what they're commissioning for genetic testing. So the leftmost panel, as you look at it, indicates the total percentage of cases harbouring one or more genetic alterations of clinical significance that is as indicated in the test directory. So then the subsequent panels is what we would look for in those genomes as well. So somatic variants, or small variants, so single nucleotide variants, insertions, deletions. You can look at copy number variants, or aberrations, structural variants, HRD, mismatch repair. And what's really cool is on the far right, you can also look at toxicity-associated DPYD mutations as well. And the really important bit there is that the percentage of tumours harbouring specific type of mutations are indicated in magenta. And that's what's listed in the test directory. And quite a lot of it is actually blue. And so therefore, there's quite a lot more that could be testing for in the NHS testing directory as well. So we were then also kind of set with this mission to kind of say, what could we return to patients? How could we improve therapy? How could we kind of precision direct those patients and make better use of that data as well? So in the last years, HRD has become quite interesting to a lot of people. So those that are deficient with HRD or HRD deficient, those patients effectively can't repair their DNA or the breaks in their DNA. And because of that, they're actually susceptible to specific type of therapy called PARP inhibitors. So we can effectively stratify our cohort of the entire tumours, all the tumour patients that we have, by the status of their HRD, so whether they're deficient or proficient. So that's HRD or HRP. But beyond that, there's also quite an interest in how to call that very well, or kind of classification tools as well. So we currently provide to researchers something called CHORD, which is from the Hartwig in Amsterdam. And we also provide HR Detect, which is from the Nick Zainal lab in Cambridge as well. But beyond that, we could also kind of classify by the genomic markers in the cohort. So we have workflows, we have pipelines in place to create cohorts based on genetic status, determine the diagnosis, survival date, and time to evaluate in terms of what the impact is for each individual or aggregated variant. And these are just some Kaplan-Meier estimates of overall survival just for KRAS and PIK3CA. So we've kind of gone through what we have seen from national registry data and the value of the genomes. But I'm a pathologist, so I was relatively selfish in kind of saying that cancer is actually extraordinarily complicated. And by just taking the genome alone or just a pathology alone, that's not quite enough for it to really expedite our understanding. We think there's a value to what we call multimodality, so actually linking pathology images, radiology scans, clinical data, and these DNA sequences, and then running what everyone's really interested in, machine learning across all of that data set. And so one of our partners that we started this program with is In Vitro, and you'll actually hear Daphne talk a little bit about the work that we're doing with them later on. We aim at some point to have probably the world's largest collection of tumour genomes linked to digital pathology images. I'd hoped it would be by the end of this year, but obviously, NHS is running a little bit slow to help us with that. So we're probably going to be around April next year. We should complete the digitisation for that. So this is my last slide. Our participants have donated an incredible repository, and there's lots of publications that are coming about the value of whole genome sequencing in cancer. We now provide a clinical service. At the moment, if you present to the NHS with specific types of tumours, you will also get whole genome sequencing. But we know that there's a need to push on further. And like Rory said at the start, how do we address some of these challenges? What do we do? So we are looking at the idea of how to utilise transcriptomics, for instance. We're looking at long-read sequencing in cancer, and could that potentially reduce the time and cost to delivering these sequencing? And also looking at the value of kind of sequencing those from diverse backgrounds. But the discussion has been about what's missing, how can we do this better? So there's a very beautiful image on the right, and I'll just kind of run through that. The process of cancer at the most simplistic level is just multiple mutational processes. And each of those processes generates a mutational signature. And these can be characterized. So smoking actually leaves a signature on the genome that we can pick up and identify. So you can see lung adenocarcinoma, and below it, it's just stacked full of red. So basically all of our participants, or most of them, have a signature associated with smoking, which is great. So I can infer, and we can infer, smoking is there. What I cannot actually say is how many of those patients actually smoked, how many of them had pack years that are more than 20 years, and actually when they started smoking and how early in their smoking career did they then ideally kind of start that tumorigenesis. And this is what the kind of ask from me or from us is that this is a bit, or the piece that's missing in this whole whole kind of catalogue. And that's where I think the next stage is to really understand very early prognostics and very early diagnoses of cancer. Potentially, primary care could advocate that for us. And that's it from me. Thank you very much for your time.